Bismillahirrahmanirrahim. Assalamu alaikum. My name is Sira Wahid and my roll number is 27. Today we are going to discuss about muscles and neuromuscular activity. There are some subtopics like types of muscles, structure of skeletal muscles, sliding filament model of muscles contraction, and contraction of skeletal muscles. First of all, what are muscles? Muscles are soft tissues. Its main function is to produce force and motion. We know that there are three types of muscles: smooth, cardiac, and skeletal muscles. There are some pictures of smooth cardiac and skeletal muscles. Smooth muscles has spindle shape, non-striated, which means that it has no bands or strips that is present in the skeletal muscles. Occurs in the walls of internal organ, uninucleate fiber, which means that it has only one nucleus on each fiber, and it is involuntary. Next is the cardiac muscles. It is striated, means that it has bands and again uninucleate fiber. It is the muscles of heart and it is involuntary. While skeletal muscles has the striated multinucleate fiber, it is attached over skeleton and is voluntary. Next is the structure of skeletal muscles. Skeletal muscle fiber has a pattern of alternate dark and light bands. Now the question arises how these bands are formed. So the answer is that these bands are due to the muscles proteins that are actin and myosin within the myofibrils. What are myofibrils? Myofibrils are many smaller thread-like structures of the muscular fiber. Each myofibril consists of thick and thin filament. Myosin occur as a thick filament and actin occur as a thin filament. Lightest region of the myofibril is the I band and the darkest region is the A band. I band contain only actin while A band contain both actin, thin filament or myosin, thick filament. Functional unit of the myofibril is the sarcomere, which extends from one Z line to another Z line. The concept of I band, A band, sarcomere, and this picture is become very clear when to see the picture in the sliding filament model of the muscles contraction. Next is the structure of the skeletal muscles. In it, you can see the muscles, the muscle fiber. Muscle fiber, in easy word, you can say it is a single cell of a muscle, blood vessels, sarcomere. Now you are familiar with this word. Sarcomere is the functional unit of the myofibril that extend from one Z line to another Z line, and the muscles proteins that are actin and myosin. Now we will see the sliding filament model of the muscles. In it you can see the Z line, the sarcomere, A band, thick and thin. Notice that the actin filament attached to the Z lines, whereas the myosin filament do not. When the sarcomere contracts, the actin filament slide over the myosin filament, and this process shortens the sarcomere and causes the muscles contraction. In it, we are facing the term like cross bridges. So, what is the cross bridges? Cross bridges is the attachment which is formed when the myosin head attached to the exposed binding site of the actin filament. It formed the cross bridges. So this is a sliding filament model of the muscles contraction. Next, we will see the contraction of the skeletal muscles. So skeletal muscle cells innervated by the motor neurons at the neuromuscular junctions. Neuromuscular junction is a chemical synapse between the motor neuron and the muscles fiber. It causes the muscles contraction. When the nerve impulse reaches at the end of the nerve fiber, it causes the synaptic vesicles to release a chemical called acetylcholine. These chemical diffuses across the neuromuscular cleft and binds with the acetylcholine receptors on the sarcolemma. Sarcolemma is the muscle's membrane. Here you can see the presynaptic membrane and the postsynaptic membrane, the synaptic cleft, and the vesicles which contain the chemical acetylcholine and the receptors that are present in the sarcolemma membrane at which the acetylcholine binds. So what will happen when the acetylcholine binds to the receptors on the sarcolemma? It alters the polarity of the sarcolemma. So altered polarity of the sarcolemma flows in a wave-like projection in the muscles fiber and make a path called the T-tubules or the transfer tubules. So endoplasmic reticulum associated with the T-tubules called the sarcoplasmic reticulum. So the altered polarity of the transfer tubules causes the sarcoplasmic reticulum to release a calcium ion which diffuses into the cytoplasm. Calcium ion binds to the troponin on the thin filament and pull it away, and then attach to the tropomyosin and separate through the actin filament also. Now the binding site on the actin filament become naked. So the myosin head attached to the actin at the exposed binding site. This binding triggers the cross bridge to bending, pulling the thin filament over the thick filament towards the center of the sarcomere, and it is powered by the ATP. Muscles contraction occur. So what will happen when the action potential stop? When the action potential stop, calcium ion is taken up by the sarcoplasmic reticulum, contraction stop, and the muscles come back to their relaxed position. Now comes to the contraction cycle. Contraction cycle, the calcium play a key role. If there is calcium present, the contraction occur. If there is no calcium present, no contraction occur. So what will happen when the nerve impulse reaches from the T tubules of the transfer tubule? So when the nerve impulse transfer or passes from the T tubules, it calls the sarcoplasmic reticulum to release the calcium ion into the cytoplasm, and these calcium ion attach to the actin filament, and troponin and the tropomyosin pull it away, and then myosin head attached to the exposed binding site of the actin filament, and this binding triggers the bending and pulling of the sarcomere towards the center and cause contraction, and it is triggered by the ATP and is powered by the energy provided by the ATP. As the myosin heads binds ATP, the cross bridge is detached from the actin, and when the action potential stops, the muscles come back to their relaxed position. Contraction cycle continues if ATP is available and calcium level in sarcoplasm is high. So it's all about my topic. Let's take the review. There are three topics of the muscles, three types of the muscles: skeletal, cardiac, and smooth. Contraction is mediated by the myosin and actin filament and triggered by the calcium ion. Thick and thin filaments, which are the muscles proteins, actin and myosin filaments, I and A band, which are the light and dark bands. Power stroke is powered by the ATP, and the calcium is taken up by the sarcoplasmic reticulum, and relaxation occur. Thank you.